Greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova. It is a privilege to be able to serve you with this after show. And thank you very much for, I'm presuming that most of you all had been a part of watching James Tour's just annihilation of Professor Dave's video. It's unbelievable what I heard Professor Dave say, and then James Tour would just absolutely annihilate everything, practically every slide of a science claim. And that's actually really disturbing that someone could perpetuate such delusional claims and pretend to be scientific. And what I'm going to have to do, it would be helpful. What I've been doing is it would be helpful to review the videos and then actually kind of go through the chemistry. And I apologize to some of the viewers who've seen some of my slides because some of it is repetitive, and I may be covering some of just the basic chemistry. If anyone would like to join who is a friend of ID and creationism, please join. Other people, let me just say this, there are other channels for that. My service is to the community of ID proponents and creationists. And people that know me know that I host opposing views, but on open mics, the quality of people who drop in have been surprisingly disappointing. And so I've left a link in the chat if you'd like to join. It should be pinned, but I'm gonna just look at this screen here and type it in again. So there are not many viewers here, and there will be an after show on logical, plausible probables channel, and that's something that I agree we should not want to miss. Besides, I have lots of other work I need to be focusing on. I did not know, and let me just point this out. I want to really thank Redefine Living for reminding me of this. Dr. Tour wrecked the clay catalysis thing. To represent what was being done in the lab is trying to represent what happens under the clays on a prebiotic earth. It was just not honest. I did not realize that. And it took someone of Dr. Tour's caliber to point that out. I might have been able to figure it out after maybe 10 years of study, but God sent us Dr. Tour to help clarify that. And I can be more confident now that those papers were not being really honest in the reporting. What do I mean by honest? Honest in the reporting would have been saying, it's with great difficulty and a lot of intelligent designed chemistry, recipes that are not at all random, that we can catalyze this RNAs on clays. That would have been honest. But to suggest that a highly scripted recipe being executed by expert chemists is representative of what would happen in a random prebiotic soup, that's just terrible. That is really terrible. And even then it doesn't work so well, which was astonishing. So probably over the next year, I will be trying to also learn some of the chemistry and teach it here on the channel. And that corresponds very well with Dr. Change Tan's book on molecular, both her book and her course on molecular biology. I've only done one lecture of hers because I've been so backlogged with other things. And I've been moving slowly. So I do invite, I know we have a small crowd tonight, but I invite my friends here to try to join the stream. Otherwise, this is going to be a very brief monologue because I don't have a lot to add. If you'd like me to repeat some things, I can do that. So say something in the chat if you want me to talk about something again. Some of you all, I'd like to thank for joining the discussion, the presentation by William Bielek, More Perfect Than We Imagined. Now, I do have one minor thing. Let's see if I can bring it up. More perfect than we imagined. Now, I do have one minor thing. Let's see if I can bring it up. Let me bring this up here. I'm sorry, I was getting a little feedback. One of my windows strangely came alive. So there's this experiment by Rebecca Blackman that was trying to show how you could autocatalytically make homochiral systems. Why bother? Why bother engineering that? I mean, carefully and not so well at that. We can prove that we actually have proof that an autocatalytic system can make homochiral amino acids or other things. We call it a living cell. We call it a living cell. What is all this trying to build and engineer a system that can do all this and yet trying to represent this as a random series of events? This is terrible science. It's dishonest science. It's fraudulent science. It's delusional science. And, well, you know, I guess that's just the way it goes these days. People want to think that they'll be able to figure everything out. And I mean, I totally get it that we'll believe things that we understand and can control. But if the miracle of life involved things that the human mind will probably never comprehend, that's just the way we might have to accept it. And it's looking to be more and more like that. So let me. Patrick Alexander said, sometimes I wonder if abiogenesis is just propaganda from the scientific community to try to get people to believe that God isn't necessary. It seems that the field of abiogenesis is kind of bankrupt. It is bankrupt. Now, I think there are some believers in God that still want to see that abiogenesis is like trying to figure out how planets orbit the solar system. They would like to resolve it in terms of natural law. And I don't have as much issue with that. But at some point, guys, if natural law is telling you that this doesn't spontaneously assemble, and that's what the chemistry and physics indicate, then you just gotta deal with the facts. So I don't, you know, I don't know that I don't have a good answer. Why is it Professor Dave is so self-delusional? Does he not watch all these videos? Is he going to just keep insisting everything he said in his stupid video is correct? I mean, this is just utterly annihilated. Utterly annihilated. And is he going to insist that this is correct after seeing this? Is he going to insist that what Dr. Tur said is false? I mean, we saw references put forward. I don't know that much about chemistry, but I could tell some of the stuff that Professor Dave was utterly stupid. It was undergraduate level stupid. In that I have a biochemistry textbook, and some of the stuff he said was just like, I can't believe you said that. He was describing a polysaccharide. He couldn't get that right. I'm like, dude, clean up your act. It's pathetic. And he was representing himself as a professor. I mean, I don't know how badly he can keep getting pounded. And that one about the supernatant proportion being all homochiral when it's only like 1%, that was pathetic. That was just totally pathetic. And he calls like a 700% excess slight. So that means you have let's see, you would have 8 parts. 1 part is like the D amino acid and 7 parts are the L amino acid. That's not a slight excess. That's an excessive excess. That's on the order of what, 87.5%. It's just Just like, dude, that's nuts. You can't be saying stuff like that and then get called out on it and just remain in denial. So this will be, he was psychoanalyzing James Tour. And, James Tour it's beneath him, and I understand it's beneath him to psychoanalyze Dave. So since I'm lower on the totem pole, I'm going to psychoanalyze Dave. Let's just monitor how long he stays in denial. I'd be very interested. Now, on the one slide Dr. Tour had there was with the phospho groups getting all messed up, where he's talking about the 2' to 5' linkage. Dr. Change Loratan saw that, referred to it in her book, The Stairway to Life, co-authored with Rob Stadler. Rob Stadler showed up on Professor Dave's channel and got booted. Professor Dave just expelled Rob Stadler, who is a scientist in great standing, Harvard, MIT, PhD, 140 some patents, millions of patients suffering heart ailments have his invention implanted in their bodies. I mean, he's a real scientist. He is a co-author with a real scientist that knows the nucleotide chemistry, and he just totally blew it. Dave totally blew it to just expel him from the channel. That's what I call denialism. That's an example. It's just beneath him. He's just always trying to save face. That's the kind of person that I see in Dave. It's more important to save face than to tell the truth. And I can kind of understand that, but something, Professor Dave is representing himself as a teacher. He has a responsibility to tell it like it is. You wouldn't want, if you have any conscience at all, if he has any conscience at all, he's not going to let his students just repeat his mistakes. If I teach something to a student and it's a mistake and it's pointed out to me, I want to do everything I can to correct it. Sometimes it's, understandably impractical because I may have sent something years ago and just like, I don't even have contact with these people anymore. But Professor Dave has a chance to set the record straight and come clean. Is he going to do it? Is he going to do it? Is he going to finally come clean? Or is his hatred of Jesus and the Christian faith so intense that he's willing to continue a lie? Because now he's been called out on it. He knows he has a mistake. At this point, to continue to perpetuate something that now he should know is blatantly false, it's no longer a mistake. It'll be a lie. And I would be curious to see what he's going to do. What is his move now? Is saving face going to be way more important than telling the truth? So, thank you for the kind words. You're a real scientist. Thank you for the kind words. I fancy myself, my official title is research associate. And I call myself a research assistant, which is a little bit less than a research associate. Because the people I had the privilege of working with, I'm just so honored. I get to work with scientists who've been studying protein biology and molecular biology and biochemistry for decades. And they've been publishing, they've been peer reviewers. I can't imagine, so many of us think about, man, I want to get my paper published in peer review. These guys sit on peer review committees. And but thank you very much for the kind words. Some of Dave's investigative reporting on the electric universe, folks, was actually quite good. But yeah, big face, lots to save. Yeah, I'm all for skepticism. So with that, if no one's going to join, I'm going to just start going into the boring scientific details, because that's, I actually started preparing something for another video, and I might as well try to start reviewing it here. let me see if I have it here. If I can actually find where I put my PowerPoint. Let me look here. Maybe in this folder on molecular biology. Okay, I have some diagrams, and at this point, just to be able to serve the viewers, I'm just going to do a little tutorial and to try to just walk people through some of the terminology. Maybe that's about all that I could do, because there was a lot of terms thrown out today, and maybe what I can do is just kind of walk you guys through it. You can type in the chat if you want, if you have questions. Think about very ignorant atheists who ate this biogenesis stuff up. Their education in critical thinking isn't very good. Thus, they not only believe the treatise, but they spread it amongst themselves. You know what? I was thinking that exactly. I had debated T-Jump, really sweet guy. I mean, I don't, he's not like Delahanty, who's really a kind of a very nasty individual in my opinion. But T-Jump's kind of a sweet guy, but he just kept repeating. I was thinking, did he learn his abiogenesis stuff from Professor Dave? Because it sure sounded like it. Yeah, this is the problem. Saving face destroys the credibility of his other videos. Whether factual or not, they will not be taken seriously. Yeah. Oh, that's all right, Patrick Alexander. I totally get it. 200 characters is not a lot. Okay, so guys, like I said, if no one joins, and I thank you for the comments in the chat, that made it easier for me to do a monologue. At least it's not quite so much a monologue if I have a chance to show stuff in the chat, so keep it coming. But in the meantime, I'm just going to walk through some chemistry to help you guys. And I feel that even if you've seen it and you've worked with it, it never helps. It never hurts to review, because even I got something out of it as I was reviewing this. I'm going to show a diagram and we'll just walk through it together, because it's the chemical terms are just going to be used over and over again. So we keep hearing about nucleotides. RNA and DNAs are made of nucleotides. Now what's appearing in yellow here is the pentose, but we can also call it the ribose. Ribose is a form of pentose. So I'm sorry that they didn't put ribose here. And you have two kinds of pentoses. One pentose is the ribose, the other is the deoxyribose. Now, does anyone want me to review how to read this chemical diagram here? Let me see if I can find. This is what they call a skeletal diagram, but also a Hayworth projection. And just to explain a little bit, whenever you see a corner in this diagram, it's a carbon atom by implication. So there's a carbon atom here, a carbon atom there, and then a carbon atom here. Now, if they actually put like an O there or another element, it means it's not a carbon. So there's a carbon here, and they call it like the 2' carbon. That's just the label to let you know what position it is on the molecule. And they have a 3' carbon, a 5'. So I believe there's 1', 2', 3'. There's a 4' carbon here, and there's a 5' carbon here. And I'm pointing this out. It can be a little intimidating. So how do we actually learn this? Okay, so when I was studying this, the professor would say, I want you to draw these diagrams on the exam. So what you do is you just spend a few hours. you get an index card and then you try to draw the diagram that you see there. See if you can memorize it. And I'd say it took me a good 2 hours to be able to draw a full nucleotide. Let me see if they get that. This is a whole nucleotide here. Nucleotide. So this is called a nucleoside, a nucleotide. They call this a nucleoside diphosphate, nucleoside triphosphate. So you see what's in blue here? It says it's a base. It's a base, and that corresponds, you have 5 possible bases. I mean, technically, you have 4 bases for DNA and 4 almost the same basis for RNA. And any questions? Let me just look at the chat here. All right. Logical, plausible, probable. You're welcome to join the chat and also advertise your after show. So we can kind of collect the gang here, and then we can send them all over to your after show. And actually, I wasn't planning on having a particularly long after show. So if you want to try to move yours up a little bit and schedule, that'd be fine. So that when we end here, we could start immediately on yours. But in any case, this is, like I said, if no one joins, I'm just going to go through this technical description. So let me look for some more pictures. I know this can sound a little rambling. So all of us have heard of adenosine triphosphate. And let me see if I can get another. I'm trying to be able to get more than one. See if I can get more than one. Okay, so good. I might be able to get more than one. Okay, so I'm going to show an adenosine. I'm going to show an adenosine triphosphate because we hear that term ATP. And now let's look at this diagram. We have this part here, which is the ribose in yellow. And you can kind of see it in this black and white. You'll see the ribose right here in yellow. It kind of terminates right around, let's see. Yeah, it terminates right here at that oxygen. Unfortunately, I can't draw a squiggly line to highlight it. Okay, so I'm gonna I think you might be able to remove your own comment. Patrick, I can try to edit out. Do you want me to, if you can count how many back, I'll try to delete them for you, Patrick. But anyway, so let me try to clear this up. So you have, this is the ribose here. If it's an OH here, it is a ribose. If you're missing the O, meaning it's deoxy, deoxy, it would be deoxyribose. So RNA and DNA, the nucleotides, the ribose and the deoxyribose are very similar. It's just, it depends on whether you have an O or not here. And I'll just emphasize that in this photo, in this other diagram. So that's why they're saying it's ribose if you have OH, it's deoxyribose if you have only an H. So again, one way to learn this, as I said, is just to start drawing the parts. And it may take a couple hours. Now the P is for phosphorus. And so when you have the P surrounded by 3 or 4 oxygens, we call it a phosphate. You can see here, these are the phosphate groups. So if you have just one P, it's a monophosphate. Two P's connected together, it's a diphosphate, and three P's is a triphosphate. Now, this here, again, to read this diagram, the N's represent nitrogen, H represents hydrogen. Anywhere you see a corner or an edge there's a carbon there, unless it's overridden by a letter, which says then it's not going to be a carbon. Oh, okay. Let me try doing that. Thank you. I didn't understand, Patrick. Thank you. All right. So I'm just pointing this out. It can be intimidating, but if you spend a couple hours just drawing stuff, and a kid can actually do this, something starts to click when you can draw it, you'll start to understand it. A single line means a single bond. Two lines means what they call a double bond. That means there are 4 electrons shared between. So there's a carbon here sharing there are 4 electrons being collectively shared by these. There's a carbon here and a carbon there. And in the case of just a single line, there's an electron that the carbon is sharing with the nitrogen, and an electron the nitrogen sharing with the carbon. So that's a little bit of organic chemistry, just to get you to see the different kinds of the representation. Now I'm going to show you the molecular representation, and if only to confuse you a little more. Yeah, so this is kind of a representation of that same molecule. So when you see this diagram here, if we could hypothetically see what the molecule might look like, if we could colorize it, it would look like that. It doesn't look exactly. So, but I mean, you could see a few things now. Usually they use the color red to indicate oxygen, like our blood. When it's oxygenated, it's red. They use black to represent carbon. The little white balls are hydrogen, and the blue is nitrogen. So you'll see this a lot. That's actually fairly customary. Someone asked, have we actually seen these connections under electron microscope? No, we have not. This is a very interesting, there's kind of a obnoxious argument I and Luca Medugno had. He's saying, we've had photographs of this. I'm just like, think again. They're actually, we've never been able to photograph this directly. We can do little kind of things that we can kind of affect the way the structure vibrates or moves or whatever when subjected to certain fields or whatever, like or you throw a laser beam through it, and we can then elucidate the structure. And like the interesting thing is we can deduce maybe the distances between the molecules indirectly, and then we can deduce the angles. But then all this is model dependent. So any photograph you see of this is a reconstruction, but not by direct viewing, either by light or electron microscopy. Now, sometimes we can do, for very large molecules, what we call X-ray crystallography. So we've not yet been able to build an electron microscope that can see this. So it is rather amazing that we can reconstruct something we can't see. This is all by inference. I've been trying to say, what's so bad about believing something you can't see directly by the evidence that's indirect? We do it all the time in chemistry. And even though now we have these nice computer-generated graphics that show the structure of the molecule, it's still by inference. It's not by direct observation. And so we're able to understand unseen realms by the observed things, by the observed realms. Of course, that does echo why many of us believe in God. We're able to understand something exists and something of God's attributes by the things he's made in the world around us? That's a great question. That is a great question. And we have more and more confidence in our models because when they predict how chemistry works, as far as we're concerned, it works well enough, and we'll keep accepting the models. So going back to the alphabet of life, this is adenine. That's the A. The alphabet of DNA is A, C, T, G. So this is the A here. This is the A. And let me see if I could find another one. The C. The C looks like that in the chemical diagram. Apologies. The chemical diagram looks like that. Now, as you start to work with this, you'll see that it's usually just a very subtle change, or sometimes not so subtle, because all these molecules look so similar. So the spelling and the shapes and structures, it can throw you off. And that's why the professor will insist you just keep practicing. And then you'll start to recognize things. And even as I was going through this, preparing for this discussion and going through all these diagrams, I said, yeah, I'm starting to see all this. So just be patient with yourself. You'll get it if you're really insistent. So I've spelled out the two bases for DNA, the A and the C. I'm going to show you the G, and it looks like that. Now I'm going to see if I could do a comparison here. Do I have one? Oh, here's a better one. Apologies. Let me see if I can make this bigger. You could see that the adenine here, okay, what I'm going to show is that all the parts here on the left are identical for each of the nucleoside phosphates. This left part, so the A, G, C, T, all the left side parts are identical. It's this part, what they call the base, that is a little different. So you could see this one has a little different drawing than this one. And the changes are subtle, but if you do it enough, you spend an hour, you'll be able to create this molecule and draw it. And then the cytosine has this shape and that one. So I'm sorry to be boring you with all this, but hopefully if you ever decide to go back to James Tour's video and he starts talking about nucleotides and ribose and phosphate groups, I hope now you'll start to feel a little more comfortable when he throws those terms. So let me see if I can find. So going back to this diagram, he had a lot of episodes where he's talking about the pentose, the ribose, and the deoxyribose and the sugar. It's the thing in yellow. And then he's talking about the phosphate groups, which are these, and then the bases. And today, he was talking about this 5' and 3' here, and that sometimes it involves the stuff that they synthesize in the lab mistakenly starts connecting 5' and 2'. Just by way of analogy, what this, sorry, I wasn't sharing my screen. Apologies. Let me share my screen. I'll try to make up for that. So hopefully, if you ever go back, my hope is if any of us are thinking about what he talked about and we want to have understanding of the terms a little better. So when he's talking about riboses and deoxyriboses and nucleotides and he used terms like these diphosphates and triphosphates. This is a good diagram. You can find it on Wikipedia. I think it was on nucleotide triphosphates, and it was kind of cute. He actually used the term nucleotide triphosphate. I mean, sometimes they'll use the word nucleoside triphosphate. I guess that could also be technically correct. But he was talking about where do you get the sugars? So this yellow part here is the sugar. And then you saw, he talks about these phosphate groups. And he was saying, there's that one scene where he was saying, oh, at 100, if you go above 112 degrees Fahrenheit, which is really, really cold, if you just get a little warmer, this phosphate group's going to just blow apart and it's gone. Or it's going to connect to the wrong part, just spontaneous degradation. So when you're hearing that, he was talking about this starting to blow apart in an RNA. See, I don't have the actual RNA here, but this is part of it. So he's talking about the phosphate group and then these sugars. So I hope that was at least a little helpful. I definitely will probably go back to the videos and try to just recapitulate a little bit of what he's saying and then do this in a more structured way. This is kind of clunky and kind of definitely ad hoc where I'm just throwing up these diagrams. I need to rehearse this a little bit better so that the presentation is a little bit smoother. And let's see if there's any let's see. So any updates? I haven't been following. I would be interested in getting reports on this. And if you came late, Jesus, I was saying I'd be interested to see if he's going to put more priority on saving face than telling the truth and saying, hey, I was wrong, because he was just obliterated. All those slides, he was just totally obliterated. And even though I'm not a chemist, I was just like, my jaw was dropping at some of the stuff he said. I'm just like, and you have supposedly a bachelor of art in chemistry. How'd you make it through school? Some of the stuff he was saying was just pathetic. I mean, if I had fellow students like that when I was going through grad school, I'd be like, dude, how did you make it? I mean, this is embarrassing stuff you're saying, and you keep repeating it. Just like when he's talking about polysaccharides, it's just like, guy, get a clue. Anyway, so that's kind of mostly what I had prepared. I didn't have anything else. If someone wanted me to talk about the problem with mixing entropy and relay synthesis, I can go over that again for some of those who missed it, because I'm really just trying to kill just a little bit of time before logical plausible probable has his after show in about an hour and a half. So otherwise I'm going to just totally run out of gas here, and there'll be kind of time for y'all to just take a break, get a cigar, so to speak. Let me see. LPP, if you could elaborate, was that Professor Dave or Professor, quote unquote, Professor Dave or Dr. Tour, quoting Science Direct? Oh yeah, oh my goodness, if Professor Dave was just quoting a Science Direct article and those are just talking points, that's really scary. Oh my goodness, that's scary. How did that, I continue to be appalled. I continue to be appalled by the kind of garbage that makes it through peer review these days. The experiments that can be replicated, that's okay. But then all these extrapolations where it's just like really funny, where Dr. Tour will say, well, how did they get that machine there? How did they get a centrifuge? How did they get a Rayonet reactor? How did they get a degassing device? And I was just laughing. I said, yeah, those experiments are real experiments, but then to represent this as a random prebiotic environment where you have random events, that is just not cool. Oh, man. I know, because he just Googled it. So LPP, we were just, I said it's not beneath me to psychoanalyze Dave. Any bets, just gentlemen bets here, whether Dave is going to just continue to try to save face or just let his falsehoods continue. Because if I made a video like that and I got called out, I would, it's like, yeah, it hurts to say I goofed up. But if you're trying to teach science students and you're just going to stick to your guns, even when you've been called out on it, yeah, I don't know. I couldn't live with myself doing that. And so I'm going to wager the guy's not going to retract. He doesn't strike me as having the sort of character that will retract. He would rather perpetuate a lie than tell the truth at this point, just to save face. He is now not acting like a unhonest broker of scientific information. His whole thing is about Professor Dave and not the truth. It's all about him. And someone told me he fits the profile of an anti-theist who, as Chris Silver pointed out, and Chris Silver is an atheist himself, but he's a professional psychologist. And the profile of an anti-theist like Professor Dave is someone who's angry, narcissistic, unagreeable, antisocial dogmatist. I mean, that wasn't me making that up. That was a very carefully conducted psychological study of that particular personality type. So I'm going to be interested to see if this plays out. And oh, by the way, greetings, awakened humanity. So if anyone has chemistry questions, if you just want me, if you had some questions about James Tour stuff, I'm happy to do what little I can to try to clarify. So what were the four major chemicals he's talking about? I was showing the nucleotides and the various kinds. You have there are all these many parts here. Why don't I just, we could, let me see if I have one. This is a nucleotide triphosphate. This is adenosine. No, is that adenosine? I think that's guanosine. Ooh, see, I'm going to correct myself here. I'm going to take my own medicine. That's not adenosine. That's guanosine triphosphate. That is a nucleotide triphosphate. Now, I just saw that some people would prefer to refer to that as a nucleoside triphosphate. But in any case, these things are the basis for making a good one here, Jesus. Oh man, that would be good. Would James Tour do a debate with Richard Dawkins? I'd pay good money to see that. It would depend on the topic. No, Richard Dawkins, I'll tell you the history here. Richard Dawkins, way back, I think in the 80s, debated also a very good chemist by the name of A.E. Wilder Smith, Arthur E. Wilder Smith. He was a 3 PhD chemist, a NATO general, Oxford. I believe he may have even had some association with Oxford. And Richard Dawkins was pretty upset after that debate, even though a small majority voted in favor of evolution after that debate. Dawkins was kind of distressed that almost half voted for creation after hearing A.E. Wilder Smith, who was a chemist of great renown at the time. And I think shortly thereafter, he might have had a few more debates with other scientists, but he didn't want anything to do with it. I think his last one was with John Lennox and people, who's a mathematician. And this could have been like, say, 10, 15 years ago. the general consensus was that Lennox won that one pretty handily. So I think Richard Dawkins has learned his lesson not to debate people like that. He won't do very well. So yes, there was someone who, a very good chemist, debated Richard Dawkins. But one thing I'll point out, debates are not, the unfortunate thing about debates, let's say that the format was 20 minutes or 10 minutes each side states its case. So Professor Dave will just state his talking points, quote unquote, Professor Dave. And then James Tour has 10 minutes to present his points. The problem is you could see that it's going to take maybe 7-8 hours of Dr. Tour cleaning up the pile of garbage that came out of Professor Dave. I mean, this is like a nasty janitorial job. So 10 minutes of spewing just garbage is going to take a 9-hour cleanup effort. And that's why I don't necessarily like debates, because some guy could just throw out so many falsehoods that it's like, where do I begin? Because everything you said, it's like every 15 seconds he said something wrong, and it's going to take me 20 minutes to explain why. And that's the problem with the debate. Someone can get up there and just state falsehood after falsehood, and you're just not going to get around to cleaning up all the mess. So that's my feeling about some of these debates. So I am glad to see Dr. Tour taking a 9-hour cleanup effort, because it's going to tell the world this is what goes on. The guys can just, one side can just spew out garbage in a short span of time, and it takes 9 hours to clean it up, to understand why it's all falsehood. Okay. They have kicked the can so far down the road that you can see the end of the road because it's light years distant. Okay. I have a request. It'd be nice to hear a bit about stereochemistry, as this is not getting talked about enough. Thank you. Okay. I have some slides on stereochemistry, and I'm trying to think. Let me, oh goodness. Someone asked, is Farina really a professor? No, he's not. He's a fake professor. And the irony was, I had a small interchange with him on his channel. I just took him to task. I said, dude, you're at the peak of Mount Stupid. You just got called out. And he said, James Tour's a liar. I wanted to respond back and say, Farina, don't you get the irony of this? You're a fake professor calling a real professor a liar. I said, don't you see the irony of this? So Farina is not really a professor. He admitted it, but kind of very obscurely. And he actually had one part where he sort of was dodgy about it. So, yeah. Custodian. Custodian. All right. So we have a request here. Stereochemistry. Okay. There are two big issues here. The one that people talk about a lot is amino acids. So there are 4 classes. I wish I had a diagram. Let me see if I could. I'm going to attempt to do a quick Google here. Okay, I'm going to see if I can get a. Oops. Let me stop sharing my screen here until I get this straightened out, because this is just going to look crazy. I'm trying to pick out something here that'll at least look half sane. All right, just to. All right, I'm just going to use a little cartoon here. And this is a great question, by the way. I want to have a productive discussion, so I might have to shrink this a little bit. So there are four big molecules that we call them macromolecules that Dr. Tour was talking about, and he had episodes that focused on each. Now, just as a caution, some people will not classify lipids as a macromolecule. Some people do. It's just easier sometimes to think of them. So four major groups. We have the nucleic acids, we have the carbohydrates, lipids, proteins, nucleic acids. There is a subtlety here because the nucleic acids have one particular monosaccharide carbohydrate that forms this. So you need this, you need at least one class of these to make this. So that's just the subtlety. But just to simplify matters, we have these four classes of molecules. This one is, they all have, they're all homochiral. They have chirality. And so we'll try to explain that. So the easiest one is the amino acids to see. And I'll try to, actually, I think I may have one that shows the nucleic acid. So this is the famous one here. This is a very famous diagram, just floats around the internet. So it's very, this, what Dr. Tour is saying, you know, you have a left hand and a right hand. You could see that they're very similar, but they're really mirror images of each other. The molecules physically in their three-dimensional configuration, one can be called left-handed and the other right-handed. So most of the amino acids that make up our protein are left-handed. So let me explain what the diagram is here. The C is for carbon, H for hydrogen, COOH is the carboxy group, NH2, that well, C is for carbon, O is for oxygen, another O is for another oxygen, and H. We call this the carboxy group. And then we have nitrogen and two hydrogens, we call that the amine group. Hence, I would presume that's why they call this an amino acid. The R is not actually a element. It is representative of a whole bunch of different possible kinds. So when you have 20 different amino acids, you'll have different R groups here. So why is this important? When, let me see. Again, this is just the amino acids. The more serious problem is with the nucleic acids, but we'll just go with this for the time being. So let me find another diagram. Let me find another diagram here. And this was actually from my biochemistry book. This is the diagram that took me 15 hours to memorize. Professor said, you'll be responsible for this on your exam. And I've often said, biochemistry is just like memorizing a phone book. It is just memorization. So I spent 15 hours learning how to draw all the amino acids. So let me see if I can kind of try to arrange this a little better. Apologies for the messy screen here. So this R group here, it corresponds to every time you see like this pink colored group here. So the R group could be any of these here, and you have just different, practically a different molecule in the R group. And that's what they meant by R group. And so when Dr. Tour today was talking about the active R groups that can connect, he was talking about, let me see if I can zoom in here. See this H2N, it could connect to another part here. It's just going to blow apart your protein. That's what he was talking about. And it's subtle. And this is why I'm trying to do this here, because without a little bit of the chemistry, he'll just mention something and it may just kind of like not really strike us. But anyone who's spent days drawing these diagrams and seeing how they connect, you're like, oh my goodness, you just blew apart the protein. And he was trying to say, yeah, that happens so naturally. So let me see where I am. Let me just stop sharing so I could find my place again. Apologies here. I'm going to check the comments. Yes. By the way, late night stream coming up in an hour and 15 minutes. So, the button peeps. So, going back to the stereochemistry, I haven't quite completed where I was headed with that as far as the amino acids. And I'm going to try to clarify that. So in the meantime, I'm going to just keep showing the button peeps because someone likes that one. So I'm doing this ad hoc on the fly, and I'm just Googling for images. So you have to forgive me if I'm stumbling here. Oh, I didn't get what that meant. Apologies. Let me show this. Sorry, Patrick, I thought you were referring to these people, these cute little molecule people. Anyway, so I'm gonna, if you can all indulge me for a little bit, I'm going to try to find something called the alpha helix. Beta, okay. See if I could find this. Trying to find a nice diagram. Yeah, here, this might be better. In fact, I used this to embarrass Professor Dave because he didn't apparently appreciate basic biochemistry here. What we have here is kind of like what we call a stick model or ball and stick model of the molecules. And then the conceptual model that is represented by a ribbon. So I mean, we would see, I mean, if we could see the connections and the nuclei and of the atoms, it would look a little bit more of a ball and stick. This ribbon diagram here, this diagram with the ribbon is more conceptual, saying that these molecules are winding around, and they actually call it a helix. This is called a helix, an alpha helix. And this relates to the stereochemistry. This is called the alpha helix. And this is called a beta strand. If they're not all L or all D, meaning if they're not homochiral, this will not form. This will not form. And if you don't have that, your proteins blow apart. Having one wrong amino acid with the wrong chirality is enough to blow this apart. That's why it's so important. And then Dr. Tour added one other twist to this, pun intended. The chiral induced spin selectivity. he was talking about that if you didn't have like this alpha helix here, that our brain would require like 10 megawatts to operate, and hence your brain would basically fry just trying to operate. Because just think about what would happen if you put your head inside of a microwave and operated at 1500 watts. That wouldn't be good. Now just imagine taking that up to 10 megawatts or 100 megawatts, an entire power plant. I mean, you'd be fried. And yesterday it just occurred to me, just to illustrate this, I think it was my sister. She worked, she was a nurse for over 40 years, maybe 45 years. And she heard a lot of stories. And there's a story of a patient that was brought into the emergency room. She was a babysitter. And the babysitter was there because she was suffering a panic attack. The kids she had been taking care of had bathed their cats. And the way they tried to dry them was to put them in the microwave, and the cats exploded, poor things. And so the babysitter had to be hospitalized because she was just in so much shock. Okay, so where was I headed with this? Our brains would be fried literally if they didn't have the homochirality. And that was something that James Tour talked about. Now, there's another aspect that Dr. Change Laura Tan discussed, and let me see if I can find that. Dr. Tan is also a very excellent scientist, Harvard trained postdoc. Let me see if I could find 224. I'm going to try to bring up my PowerPoint slide, and maybe I'm hoping I can make this a little clearer. Oh, by the way, just since I have this window up and while my PowerPoint is booting, Rebecca Blackman and who was it? Professor Dave, Professor Dave's just slobbering over her experiment that really doesn't do very well. And talking about they figured out how to get at least part of an autocatalytic reaction to make L-amino acids. I said, well, we have a model of it that makes L-amino acids an autocatalytic network of reactions. We call it a living cell. And there are also bacteria, I'll just mention this, and that many of them can take D-amino acids and convert them to L-amino acids. So there you have it. We actually have models of it. So why go through all this trouble to try to build it? You have working models. Why don't you just copy that? I'll tell you why they don't just copy that, because that look like special creation, but that might be the most successful way to do it. And Dr. Tour is pointing out, the way you guys are trying to go about it, it's just not going to work. I mean, he didn't say that explicitly, but he is giving out the chemical principles of why their autocatalytic reactions are not going to succeed, simply because they're not well designed enough. The only place it's designed well enough is in the cells that God made. Now, he didn't say that. I'm saying that. So, but I'm just extrapolating from what the data that he provided. Now, let me see. Nope, that's not the one oh dear. Let me just try one more. See if I can find one more slide. And that's why it's nice when I don't have to do a monologue. Someone can take over for me when I'm looking for stuff. But I found the slide I wanted to show. And let me make sure that this got on the StreamYard link, just so I can make sure that you're seeing what. Let me share my screen. And this is going back to the question our viewer, and that was Mr. Jetty, had some time ago about asking more about stereochemistry. All right, so what Dr. Tour was emphasizing is if these are not homochiral, this is like trying to build a railroad track with the screws going the wrong way in, when you're trying to bolt things, or no, that's not a very good analogy. I'm trying to think of a good analogy, but the idea, if you build a railroad track, you want to have everything uniform. You want to have all the parts uniform so that the track lays out nicely. And it's kind of the same idea. If you want to build a readable system, you want to have something uniform enough that it could be read. So tonight, Dr. Tur was pointing out that the proper connection, you could see this phosphate group here. It connects the 3' sugar of this nucleotide to the 5' sugar of this nucleotide. And that always repeats 3' to 5', 3' to 5'. So you have this phosphate group always connecting 3' to 5'. What happens randomly, if you don't do this, you get a connection of 3'. You get a connection of like what, 2' here to 5', and you get basically a gene killer at that point. But before I even go that far, let's see if I have a diagram. The diagram of the thumbnail we had, I'm going to have to kill some windows here so I can clean this up. And I'm going to try my best to remember my train of thought. Was the question of chirality. If you remember the thumbnail, in fact, I'm going to grab the thumbnail now that I think I'm going to grab the thumbnail, and let me look for it. And I'm going to show some chirality here. This was the thumbnail to our video, but it also shows the chirality issue. Our DNA is made of nucleotides that look like this here on the right. It's called, in fact, it identifies it as the normal, quote unquote, form. The normal D form is here. And so when they say that our DNA has 3.3 gigabases, that means you take 3.3 billion nucleotides like this and connect them together to make up our chromosomes, etc. 3.3 billion of these molecules to make a single strand of DNA. And actually it would be 6.6 to make the double helix. That's a lot. That's a lot of molecules. And that contains the genetic code plus other things. If, however, biology was trying to assemble the DNA from a mix of these, it wouldn't work. The geometry would fall apart. What would not happen, what you would not be able to build, is this nice railway. So when you see this thing here, that's, okay, so this, I'm just going to try to circle it. That's a nucleotide. That's a nucleotide. What we want is to build the DNA with this right here. If you don't build it with this, you don't get this, and it's not readable. This is just like building a railway track, and every other step or so, it's all messed up. So that was a long answer to the question, but I felt it was important because that's the importance of the chirality. You just end up not being able, it's just very simple. It's very similar to the problem of why you want uniform parts. You want all the screws to be threaded the same way, right? In the nuts, that's really nice. Because if you had one nut that threads the reverse, like when I'm trying to tighten a screw, you want it clockwise. But if you had screws that were, like say nuts that are threaded the other way, and the bolt is threaded, that just doesn't work. All right. That's the closest analogy that I could give. That's why, that's one of the reasons, in addition to what Dr. Tur had on his last lecture on CISS, chiral induced spin selectivity. So there are geometric reasons why life would fall apart. And so thank you for the question, Mr. Jetty. So I'm way behind on my reading here. And I'm just going to read through the comments. I'm just going through these. Yeah, Tony Murray's. Yes, I think Dave would make a comedian. We're getting a lot of laughs out of him. Yeah, no, I leave that to you, Tony. Jesus is king and tourist is messenger. Oh, okay. And thanks for encouraging people to support my channel with the upvotes. Oh, very important. There's a late night stream at 10:30 p.m. Central Time. That's an hour from now, and you guys want to join the stream? Otherwise, I probably will start trying to wrap this up because I am running out of gas. It is hard to run a stream like this with a monologue and doing all this ad hoc, trying to respond to questions and Googling. Yeah, how could Dave possibly? I don't think he's going to. And maybe you heard us earlier. I'm making a bet he's not going to withdraw. He's going to attempt to save face by perpetuating falsehoods. And, at this point, now that he's been called out, it's no longer just a mistake or a misunderstanding. I would classify it as an outright lie. I rarely call people liars, but he's been called out so badly on this. For him to keep insisting he's right at this point, just doesn't look good. Yeah, he could respond with an apology. Yeah, I shouldn't laugh. That's really kind of pathetic. Proteins remind me of my dog. Yeah, amen. If yes, you know what? I'll tell you what's disturbing. We may be focusing on Professor Dave, but he is representative of entire industries, both the industry of abiogenesis and the industry of evolutionary biology. They will not budge. And I saw a garbage paper on evolutionary biology just come out through the proceedings of the National Academy of Science. I said, I can't believe what they're just letting go through peer review. I mean, some of the professional scientists that I've had the privilege of knowing, they're just appalled. It's like, how do these guys get away with it? Just like, that's why you could see, it's really nice to see Dr. Tour so passionate. He said, how's this happening? This story has to stop. And it just keeps going. And it's not just about Dave Farina. It's the whole industry. It's the whole industry. Their experiments are good, but they're misrepresenting it. It's like, how can they in good conscience represent all these carefully, intelligently designed, carefully crafted recipes, chemical recipes, as representative of random events? That's just not cool, guys. That is just not cool, abiogenesis researchers. So let's see. Oh, I have a question about chemical bonds. Do double, triple require more energy to start and also maintain? Also, does it require more energy to break? Oh my goodness, if Dr. James Carter were here, who is an organic chemist, he'd be able to answer that question. But now you got my curiosity up. I think we can get a quick Google answer. Single, doubled. Okay. No, I actually don't. I actually don't know the answer. And it's really okay. This is going to be embarrassing because I'm giving a lecture from OpenStack's chemistry. I haven't gotten to this part of the OpenStack's chemistry in my Evidence Reasons Academy, where I'm giving chemistry lectures. So I just haven't gotten that point. So let me try to share my screen. And I appreciate the question. I'm glad you're asking these questions because, okay, we find that triple bonds are stronger and shorter than double bonds between the same two atoms. Likewise, double bonds are stronger and shorter than single bonds between the same two atoms. So let's pick out a diagram here. This is a single bond, and this bond is stronger. So there's, as I said, there's an implicit carbon right here. They could put a C there for clarity, but just for shorthand, they just assume a carbon at every corner. So this bond is stronger and shorter between this nitrogen and this carbon because it's a double bond. And this bond is weaker and longer. So thank you for the question. That was awesome. That was awesome. Thank you for the question. And this also requires specified electric polarity for the amino acids as well. Yes, there are some amino acids that are polar, and I should know this since I'm supposedly a protein research assistant. I need to know that a little bit more off the top of my head. There are some that are polar, and see if I've since lost that diagram. Let's see. Just let me see if I could find it. Okay. I think I found one that shows polarity. Here you go. We have some amino acids here, and they're this group. You have 1, 2, 3, 4, 5, 6 that are classified as being polar. The proline, asparagine, glutamine, cysteine, threonine, serine. There are some that are negatively charged, some that are positively charged. So those would also, there would be electric polarity there if you have one that's positively charged or negatively charged. And then we have some that are what they call nonpolar. And then we have also, we have two groups of nonpolars here. Yeah, so these all have different properties, and they're very important for being able to make proteins. Doesn't the chirality and type of bond that forms have similar relevance in relation to proteins blowing apart? Absolutely. Absolutely. I showed earlier the picture of the beta sheets, and I'm just going to bring it up again. Let me see if I can bring this one up again. Thankfully, I have it somewhat handy. This alpha helix would blow apart if you didn't have homochirality. Now, just for completeness, if it were all right-handed, you'd still be able to get an alpha helix, except the rotation would be just the reverse. But it would still, you'd still be able to get an alpha helix, and likewise for this beta sheet. Now, these are very important structures to making proteins, and maybe I could show a. Let me see if I could find a picture here. Oh. See if I could find. One of my favorite cool looking proteins is a helicase. See if I could find one. with a nice ribbon diagram. Now I'm doing this all on the fly, just trying to Google and hoping I can find a nice picture. I think I may have found one. So, and let me hide this. So you see this ribbon here? That's the alpha helice. And you have, if that blows apart, your helicase blows apart. If your helicase blows apart, your DNA is not able to replicate, or and hence you're practically dead. You're as good as dead. That's an illustration of the importance of homochirality. So let me make sure I doesn't chiral, and the type of bond form. Yes, the type of bond form, it has to be what they call an alpha peptide bond. Dr. Tour is pointing out that you can get it to bond in different ways. And he kept talking about, just like when you're, just let me back up a little bit. How many times have we been, some of us that will buy things from the store and we have to do some self-assembly. Sometimes the parts can be such that you might connect something wrong and not realize it until, oh my goodness, I wired this backward, or, you know, it can happen and it just won't work. The thing is, in a random soup where things are not controlled and things, it's just like putting glue on a bunch of Scrabble letters and then just throwing them in a pile. They're not going to spell any. It'd be just like a total mess. And what's bad is they're connected together. And so it can happen in a random soup. If you can form all these bad bonds where they don't connect, you'll end up trying to build your self-assembly thing. And it'd be like just throwing glue on all of this and just shaking it and seeing how they connect. Yeah, you'll get things that will not be good. You won't be able to form usable proteins that way. Yes, it will blow apart, so to speak. And by the way, this is why it's good to kind of go through the chemistry and then try to relate it to, you know, just kind of our own mechanical world. Because when you start to go through this, you start to see it. And that's what I feel. I have a real passion that as the ID proponents and creationists get at least a little chemistry, it'll be more than just repeating quotations. You'll actually see why with Professor Dave, you'll see it at a different level. You'll see it more and more like the way Dr. Tour is seeing it. You could start to see why he gets so angry now. Because as I started to study this, I was like, oh my goodness, I can't believe that a fraud is being committed on the public. It is so obvious. You know, the reason they have to do all these experimental steps is just like trying to assemble something. You need to get the part oriented in the right spot to get to connect to the other part in the right spot. That's why there are all these, like that 90 pages of chemical steps that didn't even work in the end. That's fundamentally why this origin of life stuff that takes 90 pages to characterize all the chemical steps, to characterize the recipe, why it's dishonest to represent that as happen in a random environment where you don't have controlled steps, where steps could be out of order. That would be really bad. And it's not honest to represent that as showing that life naturally arises. That is just not right. Anyway, I believe that would have to do with the repulsion and the wrong charge. What really blows it apart is first the chirality. If you're lacking homochirality, the next thing is if you end up getting bonds, which is not so much electric stuff that has something to do with it. It's quantum mechanical, but just think about it just like is just parts that would naturally glue together if you didn't orient it. You know, it's just like if you have Velcro or tape, and you know you can connect it, it would just so naturally attach. That's the problem. You have to orient the parts before you connect them, and that And so when Dr. Tour is talking about protect and deprotect, and he's talking about all these protection chemicals, that's to prevent the wrong part from connecting to the wrong other part. And when he was covering the problem with the 2' 5', that's what he was talking about. He said, if we're going to do this in the lab and to prevent this from happening, we have to protect and deprotect all the parts. So instead of orienting it correct, what you do is you just put a plug there. So even those random molecular motions, they'll eventually get it right. And the parts that are supposed to connect will connect, because you basically you prevented one of the connections from happening by accident. And it's tough to do that sort of chemistry. It's just really tough. Integrity doesn't exist. Magic arrows. Oh my goodness, that was great when Dr. Tour quoted Leslie Orgel and saying the autocatalysis network is just magic. Would be magic. So yes, we need to think of a better analogy at some point. Clockwise versus counterclockwise. Actually, speaking of this, we actually do use the term for alpha helices. Let me look this up. Alpha helix. Okay, I'm gonna mess this up if I don't just try to read it verbatim. Let's see what it says. The screw sense of an alpha helix can be right-handed or left-handed counterclockwise. All alpha helices in proteins are right-handed. So the alpha helix that I was showing you, which is this one, the biophysicists in the group would call this a clockwise. It is called clockwise. So obviously they have a way of defining it as rotating clockwise. It's a right-handed screw. So you have L amino acids, left-handed amino acids make a right-handed alpha helix. Okay, that's just the way it works. That may have been the conventions that they arrived at to call one thing clockwise or counterclockwise. So that term is actually used to describe the parts of the protein as being clockwise, and they're always clockwise. Almost always clockwise, because they're almost always left-handed. There are very, very special exceptions to that, but that's by deliberate action of the cellular chemistry. That has to be very well controlled. So, good question. Where can we find Dave's responses to Dr. Tour? Personally, I don't feel like digging through the garbage can of his channel. So who should be a point for this job? Will Kincaid comes to mind. I'm sorry. We have to find someone to dig through that. So save your Streamlab hours, Sal. Yeah, actually, for Dr. Tour stuff, I'm going to, since we only have, 12 point. Oh my goodness, this is sad. He might have only 12.2 and then 13. We might have only 2 more episodes. So that's why I'm spending a little extra time for this. Because then after this, I might not be doing so many live streams. It's been, by the way, just a great deal of fun to do this with you all. And because Dr. Tour is so special, I'm going a little bit out of my way to do these after shows to serve the audience who's supported him and watched his stuff. Oh, gracias. Thank you, Jesus. Little Davey is now irrelevant. Yeah, he is. He's now just our source of comedy. I know that's kind of harsh, but that's the truth. After tonight, my brain is so full it's leaking nucleotides and peptides. Yes, PNAS for short. That's Proceedings National Academy of Sciences. Sorry, I've just done so much work in the electrical field that it just made more sense for me to think from that perspective. Yes, it's nice to think in terms of positive and negative charges, and indeed at the atomic level, there's positive and negative charges. But the thing about the atomic world is you have this thing called quantum mechanics, where things bond together based on quantum mechanics, and it's just weird. We can just accept it. Sometimes we just empirically accept it, just like when we throw these two molecules, these two atoms together, they're going to form a bond, and it will result. It's going to it's going to end up being a particular shape. There have been a few people that made mention of his responses, which appear to only be ad hominem. I wanted to say, I've always been curious when Dr. Tour shows, when a video of his is posted, you'll have like 80 upvotes and then that solitary downvote. I wonder if that's Professor Dave. I wonder if he's just, speculation. He responds to a lot of comments, deletes a lot too. Yeah, he booted Rob Stadler, which was just ridiculous. He booted a real scientist, and yeah, that shows there's no integrity there, no integrity, because Rob Stadler, I mean, anyone who's watched him, he's a straight shooter. He's just going to tell you the science. He's not going after anything else. And for Dave Farina to do that, that's like, yeah, you're going to save face. And he did that, I believe, before he did his James Tour thing. I don't know that for a fact. I may have that wrong, but that was bad. I don't think he has an agenda. It's not hidden. His agenda is very anti-Christian. He calls the Bible an immoral book. He mocks Christians. He thinks that just because someone believes in the resurrection of Jesus Christ, that they're unqualified to be a scientist. So it's like, well, who would we have to disqualify? We'd have to start with like people like Isaac Newton, and then all the great scientists who are in the foundation of modern science. We could have a huge list of them. So they're not so many today compared to back then, but there's still a lot. So that's one of his drones. Yes, we at least did get a interview on Standing for Truth's channel out of that Professor Dave booting him. Yeah, that motivated him. Thank you, Zach, for your kind words. And I think that's it. I finally caught up on everything. I do plan to try to review Dr. Tour's stuff and then build lessons around it. God willing, it won't be as clunky as my ad hoc presentations today, where I'm just desperately Googling for an image. Hopefully I'll have time to look for really good images and ways to describe concepts in a way that hopefully will be more effective and faster to communicate. And there were some really good questions today about chirality and even the strength of single, double, triple bonds. I think there's a way, and I've been meaning to try to do this. We could probably not have to force people to, we could probably find a way to teach people enough organic and biochemistry that they could understand what Dr. Tour said without having to go through 3 years of a biology or chemistry program in college. I'm pretty confident that that can be done. Part of the reason for that was when I was getting my retraining in biology, I just like, oh my goodness, do I have to go through all this chemistry? And my professor said, no, you'll be able to pick it up. It'd be hard, but you'll be able to pick it up. And so I think that with a little bit of dedicated effort, meaning spending a couple hours here and there just to get the basics, like when I was walking through the chemical diagrams, you'll be able to understand all the major terms. So if you can distinguish a amino acid from a nucleotide, from a lipid, that would be great. And you'll be able to pick that up just from the diagrams rather quickly. You'll be able to see the lipids are probably the easiest to recognize as a molecule. But when you could see the parts of a nucleotide, like the ribose, the base, and then the phosphate group, that might take a couple hours to learn. But then whenever you watch these debates on abiogenesis, and when they're talking about nucleotide synthesis and DNA synthesis, you'll know who's BSing you. You'll know it intuitively because you've had 2 or 3 hours to learn the principles. You've had that background. And I think that would, there'll be a good benefit to cost in terms of the time invested. And you'll feel better about yourself too, that you've learned a little bit of organic and biochemistry so quickly. So yeah, I'm about to shut down this stream, and then we have a half hour to rest before Logical, Plausible, Probable begins his live stream. Yeah, Jesus Garcia. Yeah, he is definitely trying to defend his personal worldview with rhetoric and an argument from personal incredulity, which is what he blames Christians for in the first place. Yes, I agree. And I know we're psychoanalyzing someone now, but he's really asking for it. And yes, welcome, Zach. So, God willing, this isn't goodbye, and maybe I'll see some of you in Logical, Plausible, Probables after show. I'm very grateful to him for trying to work out the schedule so that we don't have a schedule conflict. And I may be hanging out in the chat a little bit. I do have other stuff I should be doing. I'm playing a little bit of hooky so I could do this, but I felt it was important since I think Dr. Tour's talk is pretty historic. I think on some level, this is going to be historic because he's calling out an entire industry. And, he's like that man that's in the gap, and he's just going to stand up to an entire onslaught of falsehoods. And I think God has appointed him to stand in the gap, because he certainly has the qualifications to bring attention to all this. And it's my goal, now that we also have Changed Laura Tan and Rob Stadler's book, to share this with science students, especially those obviously who are intelligent design friendly. They will be equipped. What Dr. Tour did in this series is like, okay, so now when these students read Dr. Tan and Dr. Stadler's book, they'll know someone as senior as Dr. James Tour, and then also pretty close right up there, if not his equal, would be Marcos Eberlin is backing this up. You're going to see some senior chemists backing the Dr. Tan's work. And it'd be very hard for any chemist, for any professor of chemistry on this planet to say this is all crank stuff, because they'll have to face people like Marcos Eberlin, who's trained 200 PhD chemistry students, 200 PhD scientists. He was their supervisor, 200, and then James Tour. So these guys coming forward, and now that we have the literature by Dr. Tan and Dr. Stadler, we could be rocking at this point. This is just gold. So, and God bless you, Mr. Bond, Mr. George Bond, and God bless you. And Lord willing, I'll see you definitely, at least in the chat on Logical, Plausible, Probables channel. So with that, take care. God bless you and have a good evening.